हेलो स्टूडेंट्स, होप यू आर स्टडिंग वेल एट योर होम। हम लोग बात कर रहे हैं पोस्ट फर्टिलाइजेशन इवेंट्स के बारे में। एंड जो पोस्ट फर्टिलाइजेशन इवेंट हमने अपने प्रीवियस वीडियो में डिस्कस की थी, दैट वाज डेवलपमेंट ऑफ डाइकोट एम्ब्रियो। अभी तक हमने डेवलपमेंट ऑफ डाइकोट एम्ब्रियो के बारे में बात किया है। अब आज की इस वीडियो में हम बात करेंगे डेवलपमेंट ऑफ एम्ब्रियो बट इन द केस ऑफ monocot plants. So let's start our new topic with new video, that is development of embryo in monocots. So today's topic is development of embryo in monocot. Like in the case of dicot, we discussed that after fertilization, when egg and male gamete get fused, they form a zygote. Same thing will happen here. यहाँ पे भी वही सेम काम होगा। फर्टिलाइजेशन हो रहा है, फर्टिलाइजेशन के बाद जो स्ट्रक्चर फॉर्म होगी, that structure will be zygote. So this is our zygote. This is our zygote, which is 2n or diploid in nature. This zygote will further divide to form embryo. Initially, the stages that take place in the case of dicot and monocots are almost similar, but later on we see loads of differences. Let's see what is there. In the starting, this zygote develops and it increases in size. After that, it divides. The division which take place inside this zygote will be ट्रांसवर्स डिवीजन। यहाँ पे जाइगोट के अंदर डिवीजन होना शुरू होगा। लाइक डाइकोट प्लांट्स इट आल्सो डिवाइड बाय ट्रांसवर्स डिवीजन। तो यहाँ पे भी जो डिवीजन हो रहा है वो डिवीजन ट्रांसवर्स डिवीजन होगा। बट वन थिंग दैट हैपेंस हियर इज दैट ड्यू टू दिस ट्रांसवर्स डिवीजन द सेल्स व्हिच आर फॉर्मिंग हियर आर 3 सेल्स। यहाँ पे जो सेल्स बनेगी डाइकोट एंड इस मोनोकोट एम्ब्रियो जाइगोट में डेवलपमेंट के बाद इस मोनोकोट जाइगोट में after transverse division the cells which form are 3 cells. यहाँ पे 3 cells बनेगी. These are the 3 cells which are formed here. What happens here? होता क्या है, क्यों ये 3 सेल बन रही है उसके बारे में बता दूं मैं आपको कि for in the starting it's divides and form 2 cells. पहले 2 cells ही बनेगा, but 1 cells divides one more time. एक जो सेल होगी वो एक बार दोबारा फिर से डिवाइड होगी and finally we get the 3 cell stage. So we color these cells in different colors. We will show it with different colors so that you will able to understand. This lower cell, this cell will be the basal cell. This cell which is towards the lower side, this is the basal cell. This cell which is present upside the basal cell, it's middle cell. And the cell which is present at our uppermost part, this is towards the chalazion end. We will draw it with another color. This is terminal cell. This is terminal cell. As in the case of development of dicot embryo, we discussed that we label the cells according to their pole, according to their location. जैसे कि dicot embryo के development के time पे भी मैंने आपको समझाया था कि हम क्यों किस तरीके से cells को refer करेंगे. The cells which is present towards the micropylar end, that will be basal cell. जो माइक्रोपाइलर एंड पे होगी वो सेल होगी बेसल सेल एंड द सेल व्हिच इज प्रेजेंट टुवर्ड द चालाजियन एंड विल बी terminal cell. So you have to mention the poles here also so that we can easily understand the cells. Now let's see what happens further. With one transverse division, in the starting with the transverse division, zygote will form 3 cell stages. This is 3 cells: basal cell, middle cell and terminal cell. अब क्या होता है इस जाइगोट में जो 3 सेल्स बनी है, आउट ऑफ दीज 3 सेल्स बेसल सेल डू नॉट डिवाइड फर्दर। मोनोकोट के केस में बेसल सेल आगे जाके डिवाइड नहीं होती, जैसा कि हमने डाइकोट एम्ब्रियो डेवलपमेंट में देखा था। आप उसको फिर से पिछले वीडियो में जाकर के देख सकते हैं, उसका लिंक हमारे इसी वीडियो के डिस्क्रिप्शन बॉक्स में दिया हुआ है। यू कैन सी दैट इन द केस ऑफ डाइकोट एम्ब्रियो द बेसल सेल फर्दर डिवाइड एंड इट फॉर्म्स सस्पेंसर। डाइकोट एम्ब्रियो में बेसल सेल जो है वो डिवाइड होती है और सस्पेंसर बनाती है, एक लॉन्ग स्ट्रक्चर बनाती है। बट इन द केस ऑफ मोनोकॉट दिस बेसल सेल विल नॉट डिवाइड फर्दर। बेसल सेल आगे जाके डिवाइड नहीं होगी, इट विल नॉट डिवाइड फर्दर। सो व्हाट हैपेंस, what these cells will form, let's talk about what these cells will form. The middle cell which is present here, the middle cell will form almost all part of the embryo. Embryo के जो important parts हैं, जितने भी embryo की development होनी है, वो development होगी by this middle layer. What's the structure was formed by this middle layer? This will form the plumule. Plumule will form by the middle layer, and radicle, epicotyle, and hypocotyle, and some part of suspensor also. So suspensor also formed through this middle cell. So middle cell will form almost all part of the embryo. Plumule and radicle, we write the name plumule or radicle here. यहाँ पे हमने एक बार plumule और radicle लिखा है, but later on we know that this is not called as plumule or radicle in case of monocot. आगे जाके आप देखोगे कि हम इन्हें किस दूसरे नाम से रेफर करेंगे, but till then you have to refer it as plumule and radicle. So middle cell will form plumule, radicle, epicotyle, hypocotyle, and suspensor. These are the some part of the embryo which form from the middle cell. The terminal cell, what the terminal cell will form? Terminal cell यहाँ पे क्या बनाएगी? The terminal cell will form the cotyledon. Terminal cell will form the cotyledon. In the case of monocot, they have one cotyledon. Monocots के केस में हम जानते हैं कि सिर्फ एक ही cotyledon present होता है, और वो भी बहुत thin, fleshy, बहुत thin, papery और sheet जैसा एक thin cotyledon होगा. That cotyledon will form from the terminal cell. Terminal cell will divide and it forms the cotyledon. Now let's see what happens after the further division in these cells, how it will form. As we already said, basal cell will do not divide. बेसल सेल कोई डिवीजन नहीं करेगी. जब बेसल सेल कोई डिवीजन नहीं करी, then what happens after that? Come here, let's come here. Basal cell will just increase in the size. बेसल सेल सिर्फ अपना size increase करेगी, it will become more larger. This is our basal cell, and middle cell will do division, and the division which take place in the middle cell will be in all planes. Middle cell के अंदर जो division होगा वो सभी प्लेन्स के अंदर डिवीजन होगा. मींस इट विल डिवाइड इन ऑल प्लेन्स एंड इट फॉर्म अ मास ऑफ सेल. मिडल सेल में जो डिवीजन होगा वो इस तरीके से होगा कि एक सेल्स का मास फॉर्म होगा, और इन्हीं सेल के मास से आगे जाके डिफरेंट जो पार्ट्स हमने यहाँ पे डिस्कस किए हैं, रेडिकल, प्लूम्यूल, एपिकोटाइल, हाइपोकोटाइल, वो सब पार्ट्स जो हैं वो इन्हीं सेल्स के मास से फॉर्म होंगे. दिस इज बेसल सेल एंड दिस वन इज मिडल सेल्स, एंड देन अपर मोस्ट जो ऊपर वाला पार्ट है that will also divide, and this terminal cell will divide. This is the part of terminal cell. It will form the cotyledon part. So after that, this part, how this will arrange, or they will form the different part of the embryo. Let's see how they will form. This is again the basal cell. Let's draw it with the different color so that we can identify this is the part of terminal cell. This is our terminal cell. So this is our basal cell. This is our basal cell, and upside this basal cell, the part that is formed are like this. It will divide in such a manner that it forms something like this structure. This kind of structure will form. This structure is formed due to the middle cell. This type of structure will form due to middle cell, and with this middle cell, this terminal cell will form the cotyledon, and the thin cotyledon that is present in the case of monocots plants will be present here. So this is how the embryo will look. कैसे यहाँ पे development होगा? This is the cotyledon. This is our cotyledon, and this part is the middle part. And what different parts are formed from this middle part? Let's see. This is the part which convert into epicotyle, and here the suspensor will form. Some part of this middle cell will form the suspensor. So suspensor will form from this part, and this part that is the radical part, it will cover from sheet like structure. So it will cover with sheet-like structure, and when this radical part will get sheet-like structure, it is known as coryza. Coryza. When this radical part get covered with a thin sheet-like structure, it will covered with a sheet, and after that we will call it coryza. And what will happen? This is the plumule part or upper part that will form the shoot part. This is plumule. It also get covered with the sheet, and after the covering it with the sheet, it will known as coleoptile. It will called as a coleoptile. So here we get the two new names. This structure will be coleoptile. And this is the cotyledon axis. This is the axis by which the cotyledon is joined with the embryo. This is the cotyledonary axis, and at the portion of embryonal axis, ये embryonal axis portion है. इस embryonal axis का portion जो cotyledon से above होता है, that portion is known as apicotyle, and the portion that is present below the cotyledon axis is known as hypocotyle. So we'll write it here. This is our hypocotyle, and this portion will be apicotyle. So let's see what happened till far. अभी तक क्या हुआ है? This is about the development of monocot embryo. Monocot embryo. इसमें सबसे पहले zygote. Zygote will divide. It forms 3 cells. One cell is lowermost cell will basal cell. Then upper cell, middle cell, and the uppermost cell is the terminal cells. And most of the part of the embryo are formed due to the division of middle cell. जो middle cell होगी वही most of the parts बनाएगी embryo के, like plumule, radicle, apicotyle, hypocotyle, and suspensor also. Here we can see that suspensor is forming from this part. This is the root part or radicle. This is the plumule part. So coryza and coleoptile both are forming from this middle cell, and apicotyle and hypocotyle are also forming from this middle cell. So middle cell will form all these parts that we mentioned here. This is very important. All these important parts are formed by the middle cells, and terminal cell will form only one thing, that is cotyledons. That is the reserve food material which is present inside the seed for the development of embryo, that is cotyledon. And in the case of monocot, thin पेपरी कोटिलिडन इज प्रेजेंट. तो यहाँ पे ये जो स्ट्रक्चर हमने बनाई है ये है कोटिलिडन की. अब हम एक बार देखते हैं कि इस तरीके से जो एम्ब्रियो बन रहा है, हाउ दिस एम्ब्रियो विल प्रेजेंट इनसाइड द सीड. एक्चुअल में सीड के अंदर कैसे प्रेजेंट होगा इन द केस ऑफ मोनोकोट सीड. जैसे ये एम्ब्रियो फर्दर डेवलपमेंट करता है, उसकी डेवलपमेंट के बाद दैट बेसल सेल एंड सस्पेंसर विल गेट डीजनरेट. बेसल सेल और सस्पेंसर का कोई रोल नहीं रहता, सो दे गेट डीजनरेट. ये डीजनरेट हो जाएंगे. Now let's see how it will arrange in the case of seed. If here we are taking the example of monocot seed, and the monocot seed which we are taking here is the seed of maize. This is the seed of the maize. In the case of maize seed, it is divided into 2 chambers. It is divided into 2 chambers. One chamber is larger chamber and another chamber is smaller chamber. एक chamber बड़ा होता है और एक chamber छोटा होता है. Here epithelial cells are present. This upper chamber is the endosperm. ये जो part है ये endosperm बनाता है. This is the endosperm part, and this endosperm part will divide it into 2 portions. Outer portion, this outer portion is proteinaceous in nature. ये जो outer portion होगा ये प्रोटीन का बना हुआ होता है, और इस portion को हम बोलते हैं alluron layer. This portion is known as alluron part or alluron layer. This portion is made up of protein. So this is proteinaceous part, and inside this inner endosperm is made up of starch. So this is the starchy part. This is starchy part of endosperm. So this is about the endosperm, and the embryo is present here. Here, this is the location where embryo will present. Let's see how the embryo will present here. We will make this part, that is this part. This is the middle, the part which is formed from the middle part. This is the complete embryo. Embryo will present here, like in this manner. This is embryo, and this embryo is connected with the cotyledon. Cotyledon will also present here, and this cotyledon will be thin papery cotyledon. So this is the location of embryo. This is embryo. which is present inside this monocot seed. So what we have discussed here, we discussed how the development of embryo take place inside the monocot seed. Monocot seed में, monocot plant में कैसे embryo की development होती है और कैसे embryo plant के seed के अंदर arrange रहता है, इसके बारे में हमने बात की है. अब यहाँ पे एक important question है. One important question that arises here is that endosperm is present here and that embryo is present here, and the cotyledon, we are already saying that cotyledon is a थिन पेपरी कॉटिलिडन. जो कॉटिलिडन है वो थिन है, पेपर जैसा थिन है, मींस इट डू नॉट हैव एनी रिजर्व फूड मटेरियल. इसके अंदर कोई रिजर्व फूड नहीं है, कोई न्यूट्रिएंट्स प्रेजेंट नहीं है. तो यहाँ पे इस कॉटिलिडन की जरूरत क्यों है? व्हाई दिस कॉटिलिडन इज रिक्वायर्ड हियर? क्यों इसकी कॉटिलिडन की जरूरत पड़ी? क्यों प्लांट को, क्यों एम्ब्रियो को अपने साथ कॉटिलिडन फॉर्म करना पड़ा, जबकि इसमें कोई भी न्यूट्रिएंट, कोई भी रिजर्व फूड मटेरियल अवेलेबल नहीं है? आंसर फॉर दिस क्वेश्चन इज दैट, क्या होता है, when this endosperm get digested, जब ये endosperm digest होता है, then this digested endosperm is transferred to the embryo. ये ट्रांसफर किया जाता है एम्ब्रियो को by the phytohormones and enzymes, all these things will transfer. तो ये ट्रांसफर होगा एम्ब्रियो को. जब ये एंडोस्पर्म डाइजेस्ट होने के बाद एम्ब्रियो को ट्रांसफर होता है, then first it transfer to the this part, that is cotyledon. First it will transfer to the cotyledon. ये पहले cotyledon को transfer होगा and then cotyledon will transfer the nutrients, the food material to the growing embryo. उसके बाद ये ट्रांसफर होगा ग्रोइंग एम्ब्रियो को और इस तरीके से यूटिलाइज होगा इस एम्ब्रियो की डेवलपमेंट में by using this endosperm, this nutrient material provided by the endosperm. Embryo will grow and it will form a plant. Embryo will give rise to new plants. So this is the process by which we can say that development of embryo take place in monocot plants. Here we have to take some differences also between dicot and monocot embryo development. Like in the case of dicot embryo development, हमने देखा था जब dicot embryo में development हो रहा था, वहां पे दो ही सेल बनी थी. One that is towards the micropylar end was basal cell, and the cell which is formed towards the chalazel end was the terminal cell. Dicot embryo याद है ना आपको, हमने previous video में जिसके बारे में डिस्कशन किया. सेल जो माइक्रोपाइलर एंड की तरफ थी वो थी बेसल सेल, और जो सेल टुवर्ड्स द चैलेजल एंड प्रेजेंट थी उसको हमने कहा था टर्मिनल सेल्स. ये तो था डाइकोट एम्ब्रियो के केस में, बट इन द केस ऑफ मोनोकॉट हियर 3 cells are forming. Second important difference that is present in the case of embryo development. In the case of dicot embryo development, basal cell will further divide. Basal cell further divide होती है and it forms the suspensor. वो suspensor बनाती है. And the terminal cell is the cell which forms all the part of the embryo. Like terminal cell will first form the pro-embryo, then globular embryo, and then heart-shaped and mature embryo. Means we can say all the parts of the embryo are formed from the terminal cell in the case of dicot plants. But here terminal cell is just forming a thin papery cotyledon. यहाँ पे terminal cell सिर्फ एक thin papery cotyledon form कर रही है. It's not taking part in the formation of embryo development. Means embryo के part जो है वो terminal cell से नहीं बन रहे. And the parts of the embryo are formed through the middle cell. इसको बनाने का काम कौन कर रहा है? एम्ब्रियो के सभी पार्ट्स को बनाने का काम कर रही है सिर्फ मिडल सेल. तो दीज आर द सम इंपॉर्टेंट डिफरेंसेस between development of dicot embryo and monocot embryo. After that, in the next videos, we will discuss about the seeds and fruits. That is also in your syllabus, in the +2 syllabus as well as in your +1 syllabus. Also, you studied about the seeds, their morphology and dispersion of seeds and viability of seeds, etc. We will take some important examples of the types of seeds and fruits. So keep watching, keep studying. Thank you.